So we're going to start with Annabelle Elsner. Annabelle is a rising junior, and she is majoring in biology. And she spent the summer in the Walker Lab, and she's from Eden Prairie, Minnesota. And so Annabelle, I'm going to turn it over to you.
Thank you, Sue. As Sue was saying, my name is Annabelle Elsner, and I will be presenting my project that I completed this summer in the Walker Lab, titled Dimerization as a Potential Pathogenic Mechanism for NF1. Sorry about that. All right, so neurofibromatosis, or NF1, is an autosomal dominant condition affecting 1 in 3,000 children born. It's more common than cystic fibrosis, muscular dystrophy, and Huntington's disease combined. There is currently only one FDA-approved therapy for those with NF1. Patients born with NF1 have one missing or mutated copy of the NF1 gene. 50% of cases are inherited from one of their parents, and 50% of cases appear sporadically during development. NF1 has various manifestations, making the condition difficult to study. All 4 of these patients here were born with one copy of the NF1 mutation, and over time, they developed mutations in the second allele of NF1 at different spots of their body, accounting for the various manifestations. So Jane here at the top has plexiform neurofibromas in her neck. Next to her, Rachel has cutaneous neurofibromas on her skin. Dylan below had skeletal abnormalities and tumors in his leg, causing it to have to be amputated. Next to them are twins, Adam and Neil. Adam has cranial tumors and Neil has cognitive deficits. These examples show how varied the manifestations of NF1 are, even though they were all born heterozygous for the mutation. The NF1 gene encodes the protein neurofibromin, which is widely known as a negative regulator of RAS. RAS is involved in the RAS-RAF-MEK-ERK pathway, which controls proliferation, apoptosis, differentiation, and angiogenesis. RAS is in its on state when GTP is bound, and its off state when GDP is bound. Now, RAS can naturally turn itself off, but this process is very slow. Neurofibromin accelerates this process. However, when neurofibromin is mutated, RAS remains mostly in its on state, and proliferation, apoptosis, differentiation, and angiogenesis become upregulated. In NF1 patients, over 3,000 pathogenic mutations have been identified in the NF1 gene. Of these 3,000 pathogenic mutations, 38% are missense mutations and 25% are nonsense mutations. I included the gene map here below so you can see where the location of all the mutations in the locus. Notice how there is no central location and they're all spread out throughout the entire gene. This makes the disease particularly difficult to study. So we know where these pathogenic mutations are. We're just left with a knowledge gap of what are the pathogenic mechanisms. So understanding the pathogenic mechanisms can help us develop therapies. I included the cryo-EM structure of neurofibromin here below. So you can see that it dimerizes with itself. So you have chain A from one allele and chain B from the other allele. Neurofibromin also has a gap-related domain where neurofibromin interacts with RAS. A recent study found that missense mutations affect the stability and dimerization of neurofibromin. Because of this, we hypothesized that dimerization is a pathogenic mechanism. The specific question we're trying to answer is, do missense mutations affect dimerization and cause the pathogenicity of NF1? An important method that is essential to our project is pull downs. Pull downs can be performed to purify proteins and better understand interactions. So how pull downs work is you start by lysing your cell and you have a mixture or a lysate of all of your different proteins. You then can add beads that have antibodies that are specific to your protein of interest. You can then precipitate the beads, wash away the rest of the proteins, and then elute your protein of interest so you can visualize them on an SDS page. An important part of our project is that we're adding endogenous tags to both chains and both alleles of neurofibromin. So we have the HA tag on chain A and the flag tag on chain B. So the idea behind this is that when we pull down, if dimerization occurs, when we pull down the HA tag, the flag tag is going to come with it and will be visualized on the SDS page because the HA tag, which is connected to chain A, is connected to chain B, which is connected to the flag tag. However, when dimerization does not occur, only one tag is going to be pulled down and visualized on the SDS page. This is because HA, which is connected to the chain A, is not connected to chain B, which is connected to the flag tag. So our hypothesis is that missense mutations will prevent dimerization and only one tag will be visible on the SDS page. Before my time this summer, my lab successfully performed flag prime editing in Schwann cells. They used PEI to transvect prime editing machinery into Schwann cells. They then did PCR screening and sequencing. So the next steps to the project are going to be to identify Schwann cell lines that are heterozygous for the flag tag. So that way they have an empty allele to insert the HA tag. We're then going to model missense mutations and perform pull downs. So my goal for the summer was to prepare for prime editing to be used to insert the HA tag onto the empty allele of the NF1 gene. So first what we needed to do is we needed to screen the cell lines that were prime edited for the flag insertion. So we sent them for sequencing. So Sanger sequencing works by sequencing both alleles. So if both alleles are the same or homozygous, we're going to get clean sequencing like the ones shown above. However, if they're different, the two alleles are different, we're going to get messy sequencing as shown below. So because of this, we believe that the top cell line is homozygous for the flag insertion, but the one below is a potential heterozygote for the flag insertion. All right, so once we find our potential heterozygotes for the flag insertion, we need a way to separate the two alleles so we can sequence them individually. If we find a flag tag and an NF1 tag and a wild type flag, we know that the cell line is heterozygous. So first we amplified our genes of interest, and then we did TA cloning. We also used blue-white screening to see if the insert was correctly incorporated. We also performed additional assays to screen for the insert. So what we did is we chose two primers right next to the insertion site. So if the insertion is not there, the amplicon is going to be very short and will travel further on the gel. However, if there is an insert, the primers are going to be further apart, resulting in a larger sequence or a larger amplicon that's going to travel less far on the gel. So once we find cell lines that are heterozygous for the flag insertion, we're going to use prime editing to insert the HA tag. So prime editing uses pegRNA, also called prime edited RNA, Cas9 nickase, which is a mutated form of Cas9 that only cuts one strand, and reverse transcriptase to make genome edits without double strand breaks. There are three important parts to pegRNA. First, you have the spacer sequence down here, which identifies the location of the genome edit that you want to perform. Then you have the scaffold sequence, which allows Cas9 to bind. And then the third part is the reverse transcriptase template, which allows reverse transcriptase to reverse transcribe your desired edit. So another part of my project was to create these pegRNAs. So what we needed to do is we needed to form oligoduplexes and then ligate them together. So first I modeled this in SnapGene. In red, I have my spacer sequence. In blue, I have my scaffold. And in red again, I have my reverse transcriptase template that transcribe or encodes the HA tag. So what I did is I annealed oligos to form oligoduplexes and then perform triple ligations to ligate the three parts together and make pegRNA. I then sent it for sequencing and aligned my model with my actual sequence. As you can see here, they successfully aligned and I confirmed its identity as pegRNA. So future steps are going to be to perform prime editing on flag heterozygous cell lines to insert the HA tag. We're then going to model missense mutations in dual tag cell lines. We can then perform pull downs and visualize the tags on SDS page to determine if dimerization is affected by NF1 pathogenic missense mutations. If we find that dimerization is a pathogenic mechanism, we might ask the question of does it also affect RAS signaling and in what way? I would like to thank Sue and Colleen, my PI Jim, Francisco for mentoring and teaching me, and David, Stephanie, and Tori for helping me every day. Are there any questions?
Annabelle, I have a question for you. Can you hear me?
Yes.
Okay. Is there a way to predict whether the mutations might interrupt dimerization or not before, like in order to perhaps prioritize them for the analysis that you described?
Yeah, so mutation, like the pathogenic mutations in that paper that I mentioned, they saw pathogenic mutations that are both in the GRE, so the gap-related domain, but also in the domain that like affects dimerization. So we were going to pick mutations that occur within that domain.
Great, thanks. Any other questions for Annabelle? Yep. Oh, you were going to clap. Okay, good. All right, let's clap again. Okay, good. Thanks, Annabelle.
All right, thank you guys.